Okay. Indeed. All right, here we go. Okay. Everyone on Zoom hear me? All right. Thumbs up if so. Thumbs up. Okay, good. Great. Wonderful. Well, thank you, Sam, for that introduction. Really appreciate it. And thank you all for being here today, both here in person and on Zoom. So I'm delighted to share with you all the research I've been working on over the past 5 years here at Clemson. Whether it's sharks or bony fishes, and as Sam talked about in Hickory Trail, I've always been fascinated by fish and their teeth. Not really sure why. It's just always from a very young age, I've always had this curiosity about the teeth and how they feed. And so this dissertation has really allowed me to really explore that fascination, to get at some what I think are interesting and pretty fun questions and findings I'm looking forward to sharing with you all here today. So let's go ahead and dive right into thinking about teeth. But first, I think we have to think about them within the broader context of vertebrate evolution. So there's an oft-quoted line attributed to the French naturalist Georges Cuvier as saying, "Show me your teeth and I will tell you who you are." And I think that we can see in looking at all the different types of teeth and vertebrates that are out there, why that quote makes so much sense. So we can see that teeth are important for form-function relationships, as we can see in the teeth of this crab-eater seal, using these teeth to filter krill from the water. And you can see them in the teeth of this alligator as well, which among other things, they can use their teeth to crush up hard-shelled prey like crabs. They're also really important for inferring diet in both living and extinct species. We can see that here in the teeth of this extant horse. You can compare the teeth of living herbivores, compare them to fossil species like mammoths, and you can infer that mammoths and horses were both herbivores, likely in very similar things based off of their dentition. So it's pretty clear that teeth are essential for understanding vertebrate evolution and ecology. However, most of what we know is based off of qualitative knowledge about patterns. They're largely restricted to mammals. And as Sam was saying, mammals are, of course, iconic. They're familiar to us, right? We're mammals. They make great exhibits at zoos. We can see in this carnivore here on the left and this herbivore on the right. We can infer just based off of their dentition, generally what they're eating. Another thing about their dentition is the mammals are generally renowned for having heterodont dentitions, in which tooth form and function exhibits kind of no recognizable variation along the jaw. So we can see that illustrated really well in this image on the left here. We can see from front to back these canines leading back to these malariform teeth. This pattern is generally pretty consistent across mammals, and we can see it as well in these skeletons. This carnivore on the left with its hyper-enlarged canines, its teeth on the back adapted for slicing prey material. And then this herbivore on the right, the anterior incisors used for kind of chopping plants up, and then their posterior molars used for grinding up the plant prey material prior to digestion. And so we recognize these patterns for a long time, and it's kind of led to a long observed qualitative pattern that there's a strong relationship between tooth morphology and diet in mammals. However, it's largely been qualitative, as I've mentioned before. But more recently, some advances in 3D data visualization techniques have allowed us to quantify these patterns using a metric known as dental complexity. And so this metric was pioneered in 2007 by Alistair Evans and colleagues, and they analyzed rodent and carnivorous molars to test for correlations with diet. And so by analyzing the surface of teeth and treating them basically as a topographic mountain, so you can see the teeth here, basically it takes a tooth and basically treats the peaks and crevices in the tooth as if it's kind of mountain topography, and you can generate a complexity metric from that. And I'll tell you more about this method in my first chapter, but essentially what they found was a continuation of that long observed pattern where herbivorous dentitions generally were more complex than carnivorous and other dental groups. And so this has been repeated many times since then on other groups of mammalians as well. But of course, as I've alluded to in my introduction, the group of vertebrates that I'm most interested in and that we know relatively very little about in regards to teeth are teleost fishes. So what about them? Well, teleosts are the most diverse group of vertebrates, making up nearly 50% of all known diversity. And they're also incredibly diverse in their dentitions. As you can see in this slide, we have the iconic piranhas on the left. On the right, the also iconic coral reef fish, the parrotfish, which uses its beak to kind of scrape up food off the sediment, and really anything and everything in between. So they're incredibly diverse, but we know relatively little. A lot of what we know about fish teeth is based off of descriptive and qualitative studies that have classified this diversity. But in terms of the broad scale macroevolutionary patterns with teethy fish, we don't really know much, particularly when we compare it to groups like mammals. And then we can also look at fish and wonder, are there reasons why fish dentitions might differ from mammals? One thing that may cause some differences between mammals and fish teeth is that fishes exhibit a two-jaw system. So most bony fishes, I should say, have two jaws, one used for prey capture, in which the oral jaw is used for capturing prey, bringing it into the mouth. And then they have a second set of jaws in the back of the throat used for prey processing. And so this kind of two-jaw system is much different from something like a single jaw system in mammals, where we might expect the patterns of tooth variation to differ when selection is acting on a single system versus a two-jaw system, as in fishes. So another thing with mammals is that because they exhibit these really predictable patterns, tooth homology in this group is really relatively simple to track. As you can see in this canine skull here, also this wolf skull on the right, this gibbon, you can generally track the same tooth type across very distant groups, and that can give you a very homologous means to understand how diet would be influencing that structure over long evolutionary timescales across a lot of different species. However, fishes break those rules. They show extreme variation in tooth number and position. This is a pufferfish with a single tooth fused into a beak. And then we have groups like this with hundreds of teeth along the jaws, with teeth kind of falling off the sides of their faces, fishes with long snouts, and really everything in between. And so with all this variation in tooth number and position along the jaw, it's very hard to track a homologous tooth in fishes. And so this dental homology is a challenge that has to be addressed in this group. And so that brings me to my broad outline of my dissertation. So with my 3 chapters here, to address that question of how to deal with tooth homology, I address that in part in my first chapter, where I'm investigating best practices for applying the quantitative tooth complexity metric to fishes. And in my second chapter, I expand that out into a larger data set of fishes that I scanned for my dissertation to broadly test the relationship between dental complexity and ecological function, in other words, feeding ecology in reef-associated fishes. And then I wanted to take a bit of a closer look at variation along the jaw in fishes in chapter 3 to understand if there are specific regions of the jaw that might be more adaptive relative to feeding ecology than others. So let's start with chapter 1, of course. And so this chapter, the big idea of this chapter was to provide an open source framework for quantifying fish dental complexity. And so this was recently published in Integrated Comparative Biology. So all the software here is open source, free to use. So the idea here is to have a pipeline for fish scientists, fish biologists to integrate their own questions, their own research into using this fish tooth complexity metric and apply that to their own studies. And so what I did was I took a subset of species, the damselfish, seen up here, and I kind of walked through a pipeline of how to segment the teeth from a micro-CT scan all the way down to doing the tooth complexity analysis. And so I looked at a lot of different methods for this paper for the sake of time and just for what's relevant to the other chapter of my dissertation. I'm just going to focus on 2 key findings in this study and how they relate to chapters 2 and 3. But before getting into that, let me first go into more details about what this tooth complexity metric is and what it tells us. So this metric is referred to as orientation patch count rotated, OPCR. And basically the way it works, if you take a CT scan, here we have molars from the original study that pioneered the method. When you have a scan, scans are made up of pixels. And so when you run an OPCR analysis, it essentially treats any group of 2 or more pixels in the same orientation as a patch. And so if you have 2 pixels that are in different orientation from each other, it's going to indicate that there's some change in the surface of that tooth, and that will add another patch to it. And so essentially, what this means is that having a higher patch count means you have a higher or more complex tooth. So if you were to compare this across a bunch of different species, having more patches would indicate you have a more complex tooth or row of teeth. And so a fairly straightforward way to understand complexity across your data set. Now, applying this to fishes, one thing you have to do once you scan your fish, and you segment out the region of interest. Usually for me, it was the lower jaw. You then want to get rid of anything that's not the jaw. So I was just interested in tooth surfaces. We can edit that away in one of the softwares in the pipeline. Then you can conduct the OPCR analysis. And then in this case, in the pipeline, I do this in a R statistical environment with a package called Molar. And then you can generate a patch count. So in this case, this row of fish teeth, 148 total patches. And that gives you what's called the OPCR total value, or the total complexity for this whole fish jaw. So again, I want to bring us back to that point earlier about fishes showing a lot of variation in tooth number and position along the jaw. So I think that this was a really key question I had to wrestle with during my dissertation to figure out how and if there's any effect of running OPCR analysis on all the teeth along the jaw at once or individually. And so one of the investigations I did in this chapter was do just that. So I was essentially just running OPCR on all the teeth along the jaws all at once. And then to compare the effects between the two, I would run OPCR on all the teeth individually, which, as you can tell from this animation and just from doing that with all the teeth, it can take some time. And so all the teeth are individually analyzed, and their OPCR values were then summed and then compared with the whole jaw analysis. And so this is important too, because most studies in mammals are looking at just one or two homologous teeth. And so that isn't really possible in fishes, right? So I wanted to understand what the effects were if you looked at the whole jaw all at once versus all the teeth individually analyzed. So the results here, essentially there were no differences when you run OPCR all at once versus running all the teeth individually and then summing the values together, as we can see here with this box plot. So essentially, there is no effect from doing it whole jaw or independently, which was good news for me for wanting to analyze a lot of different fish jaws in my later chapters. And so the recommendation that I provided here in the paper was that running OPCR all at once is an appropriate method for whole jaw analysis. So that was good news for me. Another thing I wanted to look at was tooth orientation. So in mammal studies, oftentimes they're focused on those molars in the back of the jaw, and oftentimes they're in a pretty standardized position. As you can see, these are just 3 jaws from a damselfish used in this study. There's a lot of variation across these jaws. You can see the curvature of the jaws and how these teeth are all kind of in different positions. And these are all just in one family of fish. So it's highly variable in terms of the positionality of these teeth on the jaw. So I wanted to see what the effect was of providing a standardized tooth position versus just keeping teeth in original functional position. So what I did was I sampled the most anterior tooth midway along the jaw, and then the posterior most tooth. And I wanted to see what the effects were when I oriented the teeth just in the regular kind of functional position and run OPCR on it. And then also running the analysis with the teeth in a standardized position where I've kind of rotated the tooth around onto the long axis lining up into a standardized position. So I wanted to see what the effects were here this way to know if there's a kind of a big change in OPCR output if you are moving the tooth position. What I found here on this box plot, so tooth complexity on the y-axis, different jaw regions on the x-axis. Between anterior and middle, there were no significant differences when you rotate the teeth from that functional position to the standardized position. However, I did pick up some significant differences between in the posterior region, particularly slightly higher with that original functional tooth position. And again, when we look at those jaws one more time, I kind of thought it makes a bit of sense seeing how some of this curvature and the kind of position of those teeth in 3D space can be highly variable, particularly along the posterior portion of the jaw. It's important to keep in mind that you can see some differences, particularly in the posterior region of the jaw when you standardize these teeth. So the recommendation I made for this study was to just take general caution in doing tooth orientation if you are doing these studies yourselves, and to really only do that if relevant to your intended study goals. For my case, I was interested in the functional position. So for everything else I present to you today, I did not reorient these teeth. I just kept them in the standard original position because I was interested in that aspect of the tooth function along the jaw. Okay, so just to generally kind of wrap up this first chapter, again, this is a paper that establishes the first general framework for OPCR on fish teeth. And my kind of broader goal with that was to make it available to other biologists to use to integrate fish tooth complexity into their own studies, particularly as there's a lot of really great work out there using micro-CT data. And I think that this method could be a really awesome complement to a lot of studies on fish trophic morphological diversification. And as I mentioned throughout this chapter, I was just looking at one group of fish, damselfishes. But for this dissertation, I was interested in a lot of other reef-associated fishes as well. So this paper provided me the framework for me to answer the questions I posed in chapters 2 and 3 also. So jumping into chapter 2, looking at the relation between dental complexity and ecological function in reef-associated fishes. For this, I broadened out my data set into 211 species across 13 families. I didn't realize how many selfies of me with organisms would make its appearance in today's talk, but yeah, I did all this at Rice University in the lab of Dr. Corey Evans. Thank you, Corey, for letting me use your facilities over the years. As you can see from these pictures progressing from left to right, this is a timeline of one of my first fishes to one of my last. Things started to get pretty, started to lose my mind a bit, I think. So Corey might not let me come back. I don't know, hopefully. But I scanned these fishes at Rice University over several years. And you can see the sampling here that I use for all the analyses in this chapter. I use the Roboski tree of life, fish tree of life. And once I had these scans, I would segment out the lower jaw. And once I have the lower jaw, I'd then have to manually isolate all the teeth. And in total, in this chapter, there were about 5,445 total teeth looked at in this data set. So going back to chapter 1, or excuse me, the introduction, I was talking about tooth complexity in mammals, right? So we know now that this has been pretty well established that mammals, when looking at their dental complexity, herbivores generally have more complex teeth than other omnivores or carnivores. This has actually also been looked at in reptiles more recently, and the same general pattern has emerged. I'll also point out that even studies on turtles, which don't technically have teeth, have shown the same general pattern that herbivorous turtles and other reptiles have more complex teeth or dental-like surfaces than omnivores and carnivores. But of course, this chapter is interested in fishes. And so with that, the kind of broad question I'm interested in this chapter is addressing if the same patterns present in largely mammals and reptiles are also found in fish dentition. So there are kind of 3 different parts of this chapter that I want to address. So the first part or question is, what is the association between dental complexity and tooth count? So in addressing this, I ran a phylogenetic generalized least squares test on my full data set. And essentially, I'm looking at the linear model results here. So tooth count, as it explains, OPCR total or tooth complexity, found a very significant effect, very strong relationship between the number of teeth and complexity, with a fairly high phylogenetic signal, just suggesting that it's a strong relationship, but phylogeny does have a strong influence here on the data. So an R-squared of 0.89 is pretty high. That's pretty strong, suggesting that about 90% of the variation in this data set can be explained by tooth counts in regards to tooth complexity. And essentially what this means is that having more teeth means you have higher OPCR patch counts, or in other words, having more teeth, you have more complex dentitions, which might make sense if you kind of think, again, as teeth as kind of topographical mountains. If you have more mountains to analyze, more teeth to analyze, you're going to have more patches present. So it generally makes sense. So another thing that I did, using the knowledge of this pretty strong relationship, I took the residuals of the fits between tooth count and tooth complexity and called that OPCR residuals in my some of my work. But just so you know, listening in, This is what I'm going to be using for tooth complexity for the rest of the talk. So taking the residuals of the linear relationship between tooth count and tooth complexity, given from this test, there's such a strong relationship between the two. So that answered the first question. There's a pretty strong relationship between complexity and tooth count. Then my other two questions are closely related. First being, does feeding ecology influence tooth count and tooth rows? And the other being, does feeding ecology influence dental complexity? So to address these questions, ran a series of tests. I ran both a non-parametric ANOVA to test for group differences, and if there were group differences, I did pairwise tests. And I also conducted a phylogenetic ANOVA using the RRPP method to assess the degree that phylogeny influences these group differences as well. And I also ran some ancestral state reconstructions of feeding ecology, tooth complexity, and tooth rows as well, just to help visualize what some of these patterns look like across my data set. So with this being a dissertation on fish teeth, I spent a lot of time thinking about how teeth are being used to feed in these coral reef environments. And so I think when thinking about this and reading a lot of literature, there's been a lot of really interesting work over the last few decades that's shown that categories like herbivory are a bit more of a catch-all term that can actually encompass numerous ecological processes. We can see that here in these different groups that I established. Typically, groups like a sediment scraper that I've assigned here, that's something like parrotfish beak, which is using it to scrape up sediment, scrape up algae and other cyanobacteria from the substrate. Algal croppers are also feeding on the substrate, but they aren't quite as contacting the substrate as much as something like a parrotfish beak. And you can see how those groups in a typical kind of classification of herbivore would kind of get, that process might get overlooked when you use these kind of broader classification schemes. And so I established these schemes to try to capture some of those different ecological processes that are often at play in coral reefs. So with these different functional categories, looking at some results here, when we look at the effect of these functional categories on tooth count, a lot going on here, but what I just want to draw your attention to here is the sediment scrapers and algal croppers. They're generally exhibiting a lot lower tooth counts relative to other groups. And once you conduct a phylogenetic ANOVA, however, I found that these group differences were no longer significant. So this suggests that while there are significant differences with sediment scrapers and algal croppers, phylogeny does have a very strong influence on these results as well. So I was looking at tooth count. When I then looked at tooth complexity, I actually found no significant differences. And even with a phylogenetic ANOVA, there's really no significant differences, no real clear effect of feeding ecology on tooth complexity across the different functional groups, which is fairly interesting. So next thing I want to do was to kind of visualize using the phylogeny and some plots exactly what might be going on there. So here on the left, we have our phylogeny colored by our different functional groups. And on the right, we have morphospace, tooth count on the x-axis, tooth complexity values on the y. And so what we're looking for here is clusters of similar colors together. So if they're clustering together, that might indicate that there's a pattern going on with tooth complexity. And what you can at least see here, stacked up on this tooth count right around 1, those are our beaked dentitions. So in this data set, I treated beaks as just a single tooth surface. And so they're all kind of aligning there right at the 1 mark. You do see some variation on the y-axis with tooth complexity as well. And then there are some outlier species ranging from some algal croppers at the top to some animal graspers and some omnifunctional groups as well. But generally speaking, everything more or less is also just clustering in the middle here. So we're not really seeing a whole lot of kind of partitioning of different groups in specific areas of this morphospace. I then wanted to look at this just by presence of tooth rows. So red here denotes just a single tooth row, meaning all teeth just align in one simple row. And then blue is multiple rows of teeth are kind of, there's multiple teeth present, as we can see in the species up here. What you can see on the x-axis is, there's a lot of spread with multiple tooth row species, which makes sense, right? More tooth rows would indicate they have more teeth. And then, of course, a lot of the single tooth rows have less teeth. But in terms of complexity on the y-axis, again, we're not seeing any you do see some kind of clustering of the parrotfishes, but otherwise we're seeing a lot of overlap between the single tooth species and the multiple tooth species. So to kind of summarize the findings here, returning to my first question, what is the association between dental complexity and tooth count? I found that tooth count is an excellent predictor of whole jaw tooth complexity, but when you then bring in tooth rows with that, I found that there was no clear difference in complexity between single and multiple tooth row dentitions once you take those into account. My second question about feeding ecology and how it influences the tooth count and tooth rows. Sediment scrapers and algal croppers were showing were shown to have less teeth, but there was a very strong phylogenetic influence on that association. And then in general, there was no clear association between feeding ecology and single versus multiple tooth rows. And then finally, my final question, kind of the broad question that this chapter was getting at, does feeding ecology influence dental complexity? The answer I kind of come to is there really were no differences in feeding ecology and dental complexity, which is really kind of surprising given what was established in mammals prior, right? Mammals were shown to generally have more complex teeth if they were herbivores relative to other groups. And so it's not really the case here in fishes. So trying to think about what might explain that. One thing that I think may be occurring here is this concept of many-to-one mapping of form to function. Essentially, it's the idea that you can have many different morphological shapes or forms doing the same general function. I think that is well illustrated in groups like the pufferfish here and the sheep's head. Pufferfish and sheep's head are both feeding on hard-shelled organisms, so they were shell crushers in my data set. And they're both using their oral jaws to acquire and crush up hard-shelled prey. Of course, the pufferfish is doing it with a single beak-like tooth dentition. And then the sheep's head has multiple heterogeneous teeth all along the jaw that it's using to do the same process. So I think many different fishes are evolving many different ways to do similar ecological functions in their environments. And of course, I think another potential thing to bear in mind is the idea of functional decoupling of oral and pharyngeal jaws that I mentioned earlier. So this has been looked at quite a lot, predominantly in cichlids, but in terms of tooth complexity, there have not really been any studies that have looked at dental complexity regarding the functional decoupling process. So I think that it's worthwhile in the future, it was beyond the scope of this dissertation, unfortunately, to look at both the oral and pharyngeal jaws, but I think a really great future direction to look at next would be what the effect is between oral jaw complexity and pharyngeal jaw complexity to understand if this decoupling process is really a driving factor for complexity between these two jaws that, generally speaking, for the most part, are doing two different things. One other thing with the kind of the application of this method that I kind of had to wrestle with a lot, there really is a lot of variation in this data set. I have species like Anamnesis cuvier, which is just one single tooth sitting on the front of its face versus having a beak dentition, like some of those pufferfishes and parrotfish, and everything in between up to a species that had about 224 total teeth along its jaw. And so the high variation in tooth count is still a bit of a challenge to address, as you have to find a way to make this method apply across all your different, when you're setting resolutions for these meshes, you have to make sure that they're all essentially equal. So that's one thing that I kind of had to wrestle with, and I think it's still been an open question on how best to deal with that and applying this method. And then I also just want to point out that there were some dentitions I simply couldn't include because they were just too weird or couldn't fit this method. Butterfly fishes, I'm sorry, Jen, couldn't be included along with angelfishes and species of comb-toothed bunnies, as my heart was sad about as well. Essentially because they have these very elongated teeth that just don't work well with the method. As you can see in this image here at the bottom, butterfly fish have these really small, interesting, tightly packed teeth, but it's that tight packing together makes it very challenging, even with a high resolution scan, to segment out those teeth individually. And so I unfortunately could not include them in this study, although I think that leaves plenty of space for future work with this method to try to solve that problem. So that was kind of the idea with chapter 2. We weren't really seeing the same patterns found in mammals applied to whole jaw studies of fish teeth. So once I kind of completed that study, the next thing I was interested in was looking at different regions of the jaw and looking at functional adaptation of dental complexity along different regions of the jaw. So again, mammals are renowned for heterodonty. In other words, tooth form and function and how it varies along the jaw. And this is widely thought to attribute to the ecological success and diversification of this group. Generally speaking, tooth complexity in mammals increases from anterior to posterior, so the less complex teeth are located towards the front, used for kind of grasping prey. And then the posterior teeth are used for grinding or slicing or kind of processing the prey prior to digestion. We're probably also familiar with that as mammals ourselves. We've all eaten. So we know what that's like, right? But it's important to point out, of course, that heterodonty is also widespread outside of mammals, and in particular, fishes. But there hasn't been a lot of work done overall looking at heterodonty in fishes, and most of the work that has been done has kind of categorized fishes into one or two categories, either heterodont with tooth variation along the jaw, or being considered homodont, which is just the same kind of morphological tooth, all positioned along the jaw. There has been some work that has started to expand beyond that. But I kind of wanted to take those discrete categories of homodonts and heterodonts and try to quantify them using this OPCR tooth complexity metric. So to get at that, the way I did this to quantify dental variation, with my CT scans, I took a measurement called the tooth-bearing bone length, essentially just the measurement of the anterior-most tooth, posterior-most tooth along the jaw. And then once I had that measurement, I then broke it up into three portional regions I could apply across my different species. And these were an anterior, middle, and posterior region. And then I ran OPCR in each of those three distinct regions to see how tooth complexity might vary along the jaw. One thing I'll point out is that I did not include beaked species in this study, given that I treated beaks as just one single continuous tooth. I thought it was perhaps moot to try to include that into this study, given that it's one single cutting edge and you're trying to partition out to different regions, just wouldn't really work for this method. So this chapter also had kind of three distinct questions. The first one being, does tooth count dental complexity show consistent variation along fish jaws? So to do this, I just did a real quick look. On our left here, we have a plot of tooth count varying across jaw region. On the right, we have tooth complexity varying with jaw region. And I did just a standard ANOVA here. And although the ANOVA was marginally significant, showing some small differences between the middle and posterior region for tooth counts, tooth complexity really didn't show any major group differences across jaw regions. So overall, tooth count and complexity are really showing little significant variation across these different jaw regions. And so that was looking at just the whole jaw and comparing things all together. So that was just kind of a first big picture question, just to get a sense of what we're doing. I was then curious if feeding ecology influences pattern of tooth counts, tooth complexity along the oral jaws, this time by taking those functional categories I use in chapter 2 again. And this time I applied them to the different anterior, middle, and posterior jaw regions, just looking at each region independently, applying these different categories just to see what the pattern looks like and kind of break things up. And so looking at this table, this is just a table showing, again, just a standard ANOVA results, and then on the right, the phylogenetic ANOVA results, this time just looking at tooth count. And when looking at the different jaw regions, I did find the feeding ecology varies with tooth count in both the anterior and middle jaw regions, although not the posterior. And in the middle region, both the standard and phylogenetic ANOVA result were significant, suggesting that there really is a very strong influence of feeding ecology on tooth counts, particularly in the middle jaw region. When looking at tooth complexity, all the standard ANOVAs for the different jaw regions came back with non-significant results. However, the anterior region, the phyllo ANOVA was significant, so that does suggest that there is, again, some influence there of tooth complexity, mostly just tooth complexity being influenced by feeding ecology in that anterior region. So how did different answers between tooth count and tooth complexity? So I'm seeing some patterns here. What I wanted to do once I had these established kind of broad patterns was then apply an evolutionary model fitting process to kind of compare across different modes of evolution to see how they might be kind of influencing tooth count, tooth complexity across the different regions. And so I did this by doing running some stochastic character maps using the make SimMap function phi tools. So I generated 500 independent trees mapping the different tooth functional categories across different jaw regions. And I applied a model fitting framework using our package out. So what I did here was I fit 5 evolutionary models fitting to both tooth count and tooth complexity. And the first 2 models, one being a single rate Brownian motion model. This is essentially with Brownian motion, it's just a model in which variance proportional to time. It's kind of considered a random walk model. And then a single peak OU or Ornstein-Uhlenbeck model. It's just a Brownian motion model with a selection parameter that's pulling the trait towards just one adaptive optima. However, both of these models with Brownian motion and just pulling towards one optima, they essentially have no effect of feeding ecology. The other models I ran do have an effect of feeding ecology, and essentially this Brownian motion multi is just affecting the rates of evolution between the different feeding ecology groups. Multi-peak is just varying different peaks that they're traveling towards, and then a variable rates model is varying both rates of evolution and the peak they're evolving towards. So looking at some results of the different models that I fit here, we're looking at AIC weights table here. What we're looking for are values that are ideally above 95 or greater, and so that indicates it's a very strong model fit. And what I found for the anterior middle region was that both were fit by models that do show an effect of feeding ecology. Anterior was fit by the Brownian motion multi-rate model, and then the middle region was fit by the OUMV model with both optima and rates varying. However, the posterior region was best fit by just a single peak OU model, which is a model that doesn't have an effect of feeding ecology. And so for results, to look at some of the parameters here, I'm not going to be showing the results. So just looking at results for anterior and middle region tooth counts. So let's look at some of the parameter estimates here for the anterior region. So this is looking at the rates of evolution. And what is clear here is that the omnifunctional species are showing the highest rates of tooth count evolution relative to other groups. Everything else is showing a fairly low rates relative to the omnifunction. So they're evolving variation between tooth counts rather rapidly. Then looking at the middle regions, again, I think omnifunctional species are here, they're showing the highest tooth count optima. So they have the highest value here relative to the other groups. And then they show the highest stationary variance. So stationary variance is more just a kind of the movements around the optima, the rate of movements around that. So showing relatively high variance and also having the highest counts there, followed by shell crushers having a fairly high count, fairly low stationary variance. So I was looking at tooth count, seeing pretty different models of evolution fitting a tooth count across the different jaw regions. And then looking at tooth complexity, what I found here was that the best fitting model for all 3 regions was actually the OUMV model, which allows for, again, both variation rates and different optima across the different feeding ecologies. So here, looking at first the anterior region, what I want to draw your attention to here is that shell crushers appear to show the tightest clustering around the highest optima here. So it exhibits the highest optima. Then we look at the stationary variance on the right. It's a pretty low value, meaning that there's pretty low movements around that optima. It's a pretty tight association. Really, shell crusher showing some of the most complex teeth consistently across these different groups. And then looking at the middle region, what we're seeing here now is that algal croppers are starting to show some of the highest complexity, although they still have a pretty weak clustering around the optima, a lot of kind of movements around that optima occurring there. And in the posterior region, again, I'm seeing that algal croppers are showing the highest tooth complexity optima, although there's pretty weak clustering around, so a lot of kind of variation surrounding there. So what we're seeing here is that there's a pattern of shell crushers exhibiting most tooth complexity in the anterior region, and then as you get to the middle and posterior, we start to see a shift in which our algal croppers are showing pretty more highly complex incisions and breaking into jaw regions. And so to summarize these findings here, with my first question, does tooth count and complexity show consistent variation along the jaws? I think between jaw regions, when you just kind of do a broad comparison, you actually see kind of minimal variation in tooth count and complexity. But once you kind of actually break into the individual regions and do comparisons within and look at how feeding ecology influences patterns of tooth count and complexity, within the jaw regions, I was finding that anterior middle tooth counts vary quite a bit with feeding ecology. However, the posterior region doesn't show any variation with feeding ecology, so it's really not under, doesn't seem like tooth count is under selection in terms of its relationship to feeding ecology in the posterior jaw region. So highly variable modes of evolution across the jaw. And then tooth complexity was best fit by multi-peak, multi-rate evolution across the jaw. So in other words, feeding ecology does still seem to have a pretty strong influence in different jaw regions, suggesting that these complexity is showing high variation depending on where you're looking at on the fish's jaw. So going back to thinking about chapter 2 with the whole jaw approach versus this regional approach, I think that it's interesting I'm getting some pretty different results here. So when you think about fish jaws, oral jaws at least, as a whole unit, there's some patterns there that aren't really coming out. Whereas if you look at, you take a heterodonty approach and think about teeth, how they vary along the jaws, you actually get some pretty different results. I think it's pretty clear that heterodonty is having a pretty clear effect on fish dental diversification. I think that's important to think about in future studies of fish teeth into the future when we're kind of continuing to do this work and consider what the kind of implications are when you're thinking about all the teeth on the jaw at once versus partitioning teeth along the jaws. And then one thing that's I want to point out, although they use very different methods in this study, algal croppers in my study exhibiting higher tooth counts, I do think was supported by some work in the Australasian grunters. So there's a smaller group of fishes. Again, they use a different method. They took a multivariate approach, including some measures of tooth shape. What they found was that in their study, herbivorous species generally had higher optima carnivores or omnivores. And so I think that's also generally supported by my study here with algal croppers as well. Going back to that result with the higher anterior jaw tooth complexity and the shell crushers, I think that's being driven by what's been deemed in the literature as spatulate tooth shape. Oftentimes, the shell crushers in my study looked a lot like this sheep's head again. I had several fishes in the family Asperidae here that often exhibited that kind of cuspid or spatulate tooth shape on the anterior region, then more kind of simpler teeth, malariform teeth position on the posterior. So I think that's generally where, generally explaining what's going on with that pattern of more complex shell crushers anteriorly. And then one other final note is that faster rates of tooth count evolution and on the functional species, I think actually contradicts some of the research on trophic specialization that's been done in some other studies of fish body shape, particularly phenotypic evolution. And I think part of what may be going on here is we can see here from two just example omnifunctional species on the top. We have chisel tooth wrasse. You can see it has that kind of similar spatulate tooth shape, but also some ridge-like teeth on the posterior portion of the jaw. And then this other species, the Mazina nibbler here, quite a bit of variation between anterior and posterior jaw there. I do think, and one kind of limitation of some of these functional categories that I've been using in this dissertation, lack of tooth functional performance data does impose some real limitations to establishing these tooth eco-functional categories. So I think that really warrants future studies that are kind of looking at how teeth are functioning in their explicit environment and really think about precisely what the function is when this fish is feeding to better understand and better inform our categorizations. And so that kind of brings me back to the whole dissertation. So to summarize what this dissertation was about. This dissertation, I think, has provided the means to analyze fish tooth complexity using data derived from micro CT scans and open source framework. In my second chapter, I applied this framework to over 200 species of reef-associated fishes, and I found that analyzing whole jaw tooth complexity in fishes actually kind of bucks the trends previously established in mammals and other terrestrial vertebrates. My results instead suggest that reef fishes have evolved many different morphological means to perform similar ecological functions. However, in chapter 3, I found that explicitly taking into account heterodonty or variation along the jaw revealed pretty high levels of variation in both tooth count and tooth complexity. And they didn't really show the same consistent patterns found in mammals, right? There's a lot of complexity in the anterior region, less so in the posterior regions, and tooth count and complexity, I should say. And so that really is quite different from what we've seen previously in mammals and terrestrial vertebrates. And so taken together, I think that these results are suggesting that fishes are making their own waves in the world of fish teeth. and they really warrant much more attention they received previously. I think additional work investigating the pharyngeal jaws would be a good next step, and would also be required to address that question of functional decoupling between the oral and pharyngeal jaws, and also looking at freshwater fishes. So this data says entirely marine fishes in the coral reef environment, but of course there are plenty of freshwater fishes doing really wild, interesting things with their teeth as well. So I think that's another direction that this can really go. However, I think more broadly, this dissertation enhances our knowledge of ecomorphological diversification in vertebrates. That's something I think, or I hope that my family is happy about. I know that young Keiffer is probably happy. From a very young age, I spent time going to museums. What I'm building up to is this dissertation is dedicated to my mom for kind of instilling in me a sense of appreciation for the natural world and fascination with marine life. Ever since I was a kid, taking me to museums and kind of well, not a good crier. Thank you, Mom, for taking me to museums and aquariums and kind of instilling this sense of wonderment in my life about marine life, from fishes, my early days as being a scientific illustrator, drawing up the body of a great white shark and, kind of really instilling in me this appreciation for the natural world. So this dissertation is dedicated to my mother. Thank you for always supporting me. And yeah. And I also want to thank everyone else, particularly the communities I've established both at Clemson and at Minnesota. But first, I want to thank my advisor, Sam. Thank you, Sam, for just letting me come into the lab and telling all these things about fish teeth and getting you excited about non-mammalian groups. Thank you for being very supportive, creating a great open environment where ideas were, even crazy ideas or ones that maybe need some more work, were encouraged and was allowed to kind of pursue the questions that really interest me and really get at this question of why fish teeth. Thank you for always pushing me to think why and broader. I know if it was a perfect world, I'd probably be just segmenting teeth in a basement somewhere doing histology nonstop. But putting it in context is always essential. Thank you also to my master's advisor, Andrew Simons, for taking me in as a neophyte ichthyologist and for giving me a chance to do some histology. And then the broader Clemson community. Thank you to everyone in the department for the many years of fun and support, even through COVID years. Excuse me, the COVID years. My committee, Rick, Kara, and Corey. Corey. Thanks for doubly being on my master's committee as well. I will say it's a shame that we couldn't ever pull off a win as graduate students as a kickball tournament, but that's I guess I'll have to live with that for the rest of my life. And everyone else in the Price Lab, thank you for all the great conversations and many fun nights and fun experiences, measuring fish, talking about fish, and just being a good support network. And then also my family, thank you to Audrey for always being supportive as well of all that I do and for being flexible, moving to many different states with me. So we're moving to another one soon after this. Appreciate all the support and for always being very willing to roll with the changes. And all my family and extended friends outside of Clemson. It really does take a village, so to speak, to support someone as they pursue their weird interests in fish teeth. So thank you, everyone. Your love and support means a lot. And then just some general acknowledgments. So none of this work is possible without a lot of different people. I want to particularly point out my creative inquiry team of students who helped me do particularly chapter 2, a lot of the data collection for that work, and also all the museum curators and collection managers. Museums are essential to all that we do as organismal biologists. And so without this work, I wouldn't be standing here. And also Dr. Keegan Melstrom for really all his excellent help and guidance when running OPCR. Really appreciate it. The Evans Lab for hosting me for all those summers in Houston. Thank you for all the help and support with running the CT scan. With that, I'd be happy to take any questions. Any Questions on Zoom? I promise I won't cry through them.
Emily has one.
Emily. So Emily, hi Keiffer, great talk. Sorry, no one can see that. Let me. So Emily says, for your results where you found a strong relationship between tooth count versus complexity, have you considered taking an average tooth complexity for a single tooth instead of looking at all teeth as a whole? Yes. So part of the method and things I explored in that first chapter were to look at the effect of running everything individually on one tooth versus, or actually just running a single tooth versus the whole jaw and seeing what that effect is. I also tested the OPCR average, which is taking all the teeth along the jaw and basically averaging. If you have like 10 teeth and your complexity is 100, you just average across that. And so that kind of provides an even distribution of complexity across the tooth. And then the chapter one, that paper kind of compares and contrasts the differences between the two and when you may or may not want to use one. But yeah, so I did kind of look at a average complexity there. Oh, I said I yeah.
Single versus multi-row. Rob, you had just the row in the book.
Hmm.
There are several, and it might just be the few, because I'm not looking at issues. I feel it in I mean, this is
Yeah,
classic, by the way. But I was thinking about it, you have the, it looks like back here,
Mhm.
that the ones that are multi-speed, right? There are some, and again, it might just be the ones themselves. Have so many fishes out there. They have a single row on the outside and then multiple rows, smaller nuggets in lining the inner jaw.
Yes.
And just curiosity wise, so you have tooth counts.
Mhm.
A lot of the tooth count might be driven by this nuggets. You've touched on it, mentioned heavy, most of those things, potentially. Again, not counting some of the entire literature of that, but have you looked at complexity of just that outer single, or it might be a little more than just single leg, that single outer jaw tooth line, kind of excluding nerve, knowing that they're there, but looking at that in terms of possibly being and thinking biologically bites. Those are bites versus I don't think those numbers are feeling.
Yes.
I'm
Yeah.
thinking sharp, not sharp.
Yeah.
I realize that it's like they're not replacement teeth as far as I very largely know. Have you thought about that? Have you done that?
Yeah, so that was a great question. So somebody in the room was asking about kind of teeth that are noticing that some of the teeth along the jaw are more kind of positioned to the exterior portion of it versus some of the more teeth in those scans that were more kind of on the interior side. And if I understand correctly, partitioning just those outer teeth. Yeah, yeah. Okay. What's that? Oh, never. Okay. Here.
Yeah. So
Yeah.
that's not the best example of it, but it's there were some that actually looked like they had almost a million, you know, bridge and then lower valve.
Yeah.
Those a little bit like there's some
Yes.
more exaggerated on.
So you're asking if like looking at just like the outer edge, more lateral aspect? Yeah.
Like sidewalk in that. They're clearly like like crushing plates.
Yeah.
There were some that like it looked like really tall teeth and then lower just nubbins right next to it. So it's more of those that maybe that was maybe your functional idea there with numbers of teeth
Yes.
that are they're literally doing something they should be doing something different like that function theme that you have.
Yes, and I think that the question, at least in chapter 3, what I was trying to get out of that was there does seem to be a lot of dentitions where there's like, some teeth are larger and more kind of complex. So just looking at them seem to be like, more lateral or more oriented towards the edge. And there's a bunch of stuff going on inside. So I've always been interested in trying to understand, how to get at how exactly is selection acting on the seed, but they just kind of, is it strong selection? Are they actively, relating to what the feeding function is, or are they just kind of there,
Right.
under pretty loose? Yeah. Yeah, so I think, at least in chapter 3, there's a lot to be done, a lot I want to kind of think through some more. But yeah, I think that trying to understand precisely and getting at, those studies of form function, if I could just, I had a perfect scenario, it would just be feeding fish and watching them, how their teeth are working, and yeah, that'd be the dream. Yeah, absolutely. Yeah, yeah, I can. I can.
This slide is perfect, I think. No, I definitely got it.
Cool.
So like this is really interesting, about the spatulate team, because I was wondering like. What is, I guess like I wouldn't gone into it under the expectation that these shell pressures would have had. Like the high complexity like at the front.
Mm-hmm.
It's starting things that doesn't happen at the front.
Right, yeah.
But I mean, it looks like spatulate at the front might be like essentially functioning to like retain the shell in the mouth, which is very cool. And then you had another result about the algal scrapers having high complexity in the middle trauma.
Yes.
So like, what do you think is going on there? Because I remember we had conversations about like looking across the jaw.
Mhm.
And, at least for me, I think algal peppers are high.
So I think at least in terms of the first point about the spatulate teeth here being more complex, I think one thing that's with this group of spirits, they're actually kind of breaking the rules a bit with that two jaw system. They are actually processing, they are grasping the prey, and they are chewing it a bit. So it's a little unclear precisely how the frontal jaws relate to that. So they do violate the pattern a bit. So I think that's part of what may be going on. They're a little more complicated. But the question about your second question about algal croppers showing kind of more complex teeth in the middle region. Um, I think in its results I couldn't include in this presentation for the sake of time. But essentially with the kind of proportion of where those teeth are, generally the algal croppers are showing less teeth. And so you have less teeth along the jaw. And because of that, there's still pretty complex, less teeth, but they're generally more complex when they're using them to feed. And so it's more so the kind of jaw proportions, I think, influencing that, the measurements of complexity. You're getting kind of 2 to 3 teeth in each of the regions, and each of those teeth is still reasonably complex relative to other groups. So I think it's more so a bit of an influence of the actual jaw dimensions.
So like it's, they're almost like okay, so this the second part of my question is about kind of that, but previously I think in chapter 2 we were looking at residuals, like residual complexity.
Mhm.
We could also, it sounds like maybe looking at the residuals would kind of get at this like thing that you're describing, or that no.
Yes, so the residuals were also used for this study as well. So so all the tooth complexity results were still the OPCR residuals taken that time I partitioned the different regions, the linear model fit between tooth count and complexity, and did it anterior, middle, posterior, and still use that study. So still kind of showing consistent results.
Still speak of the other, yeah.
I didn't partition the specific residuals for that group, at least in terms of like just looking at that. Well, I pulled the residuals, but I didn't look as closely. So I think that's good. Good place to look. Yeah. Thank you. That's any- Yeah. Good question.
Question.
So, Corey, how do you think functional heterodonty plays a role in the tooth complexity of these reef fishes? Excellent question. It's been something I've been wanting to try to kind of pursue in the various points on and off. I think so. Depending on, I think a lot it's going to have to do with what kind of functional group you're looking at. I do think that if you're approaching it from a classic homodonty perspective and applying that idea that you have morphologically discrete homodont fish teeth, and you look at their complexity across the jaw, my best guess right now is you may see pretty similar complexity values, given that the functional heterodonty, functional homodonty continuum has been applied in mostly homodont piscivorous fish. So you may not see as much complexity varying there. It may have more to do in that case with the actual lever mechanics or forces being exerted on the teeth. But I don't know. That's always a project I was waiting to stop and start with Carly Cohen and Jonathan Huey. So stay tuned. That's it's always on the periphery. Yeah.
I have a question about like fray retainment. So a lot of your functional groups can kind of like act with feeding, so like biting, brushing, nipping.
Yeah.
But I know in some like suction feeding piscivores, the prey captured by suction. And they'll have like large, patches of really small like milliform teeth.
Yes.
That are just used for like a frictional surface, the prey coming back out. Closely.
Mhm.
I was wondering if there's any teeth you see that are more used for like prey retention, keeping it, getting it back out rather than, actually participating after the process.
Right. And there has been some really cool work done specifically on piscivores recently that has looked at that, like actually finding teeth that are on specific portions of the jaw that are functionally used for either kind of strictly piercing the prey versus more kind of like holding in place like villiform. I think with the sampling that I use, I tried to be a little more selective and go for more kind of classic ram biting fishes and not do as much with the villiform dentitions, in part because again, a lot of the villiform teeth being pretty small and segmenting issues with those as well as kind of trying to keep it close just to the biting group. But yeah, I do think that could potentially play a role and potentially with looking at more kind of suction feeding biters, that could be a interesting next step. If I can crack the best way to apply a bunch of tiny teeth and analyze them all at once. Yeah, maybe you could help me with that. Add another chapter to your dissertation. Sure. Okay, well.
Thank you very much, Keiffer.
Thank you, everyone on Zoom. I very much appreciate you coming. I think we're going to take a break, but thank you again, everyone. I really appreciate it.
I started control. Sultans. Well, I was thinking, like, just make the deal. I love it. I'll do that. I know lots of. The Joe Spirit microphone synergy. So I'm so. I'm so afraid. Yeah. Yeah.
No, yeah. I told you this, I think it's getting at the functional.
Exactly. What is it? And because like the buildings, they're like little ice scrapers, they have like huge, they're very frustrated, enormous teeth. Why do they have that? They don't need them.
I know.
And mine were still good. Yeah, it's like personal.
Yeah, whole word. I don't see how we're not.
It's very. Humans are like, I'm putting on phones.
Yeah, so I kind of had to collapse. Technically, it's like browsing. Collapse small, it's not cropping, but basically defining. Really aggressive. Point about stuff. Grab it, decide one. Yeah.
Asking about the.
I was like,
I was waiting. Yeah, but like the upper lower jaw, they're totally, totally.
yeah, like that, like the fidgeting, the operating on the possible second machine.
No, and in my early days. It's cool, but there's lots of. A lot of work the same, just like.
I think it's like differences in you're looking at like when you're in looking at the scan, figure out.
Yeah, like how often it just like all from it, you know, whether upwards, lowers, like, but they commonly knows to it.
I think there's a lot of variation. I'm listening about that. There's if you count, even. Very nice. Speed, bang down the sides. Approximately. Yeah, yeah, it's like that. Yeah, especially.